Hi, I'm Deirdre Loughnane. I'm a PhD candidate in the Temporal Ecology Lab here at UBC in the Forest and Conservation Science Department.
And I'm Lizzie Wolkovich. I'm an Associate Professor of Forest and Conservation Sciences in the Faculty of Forestry at UBC, and I run the Temporal Ecology Lab. Sure. So the Temporal Ecology Lab is really interested in studying the impacts of climate change and how climate change changes plant communities themselves. And so we do that primarily right now through the lens of phenology. Phenology is the timing of recurring life history events. For things like plants, that's especially leaf out, flowering, when they produce fruit and seed. That's the full, growing and reproducing for plants. Basically, there's all these temporal events that happen. And what might happen with climate change, because already we know that most temperate plants are shifting phenologically a lot with warming. So the estimate is 3 to 6 days per degree Celsius. Here in Canada, we've warmed a degree or more. But that's the average. Lots of plants are moving earlier, a week or two, especially spring plants and spring events like leaf out. So we know there are these dramatic changes where when spring starts for plants is shifting a lot. We know it's changing depending on which event. And it's also changing tremendously depending on which species you look at. So within a community of trees, for example, what we focus on, you'll see that some tree species are shifting a lot and some are shifting less. Usually it looks like the early ones shift a lot more, and something like a birch, whereas an oak, for example, might not be shifting as much. And what we're interested in is because we know that for the most part, for the last 10 to 12,000 years, climate has been stationary, that there's been sort of a stability in the climate. So when these plants leaf out and how they sort of assemble as a community temporally has been rather consistent. We're interested in knowing what happens when it changes dramatically. Will there be some species, potentially those that are shifting the most, that are the winners, as we might say? And is it bad if you're not shifting a lot? We have some evidence. We see that with invasive species, a lot of plant invaders tend to be those that shift more phenologically compared to native species. So that suggests that maybe it really matters to how communities assemble. And we know there's sort of other correlates when we look at warming experiments where it looks like performance is enhanced by species that also shift a lot. So we have a lot of hints that maybe this matters and that maybe it has knock-on effects where some species will do better in the future and start to dominate communities and others will fare worse. And that's what we're really interested in. So we started studying that. I started here at UBC 4 years ago, and when I came, I wanted to look at this across a gradient across BC, with our most southern site being Manning Park, and actually our most northern site is Smithers, BC, which is a different regional park, Babine Mountain Regional Park. And wanted to just look at what the plant community was doing, look at the trees, see how they assemble, and see how they phenologically might be different. Within one small place like Manning Park, we have 20 or more woody species coexisting. We think they have different phenologies. We want to understand how much it matters to community assembly.
Yeah, and for my PhD, I have been essentially collecting some of the foundational data we need to understand these processes. For woody plants, we know that there are 3 main environmental cues that influence bud burst. It's winter temperatures, spring temperatures, and day length or photoperiod. And so my work entailed going out to Manning Park and tagging individuals and collecting samples, and then doing a growth chamber experiment where I controlled those 3 factors. And then we have a better sense of which species are dependent on which factors and how these might actually interact, or the complexity that we might see across species. And then as a second part, to provide a more mechanistic understanding, I'm also interested in tying in other functional traits. Phenology is just one of several functional traits that we've studied, but often it's not included in the broader framework of functional trait ecology. And so my research is trying to look at the associations between phenology and other key traits that we understand in terms of the growth strategies of plants, in terms of the competitive abilities, and tie all that together to try to understand if we can predict future changes.
Which is a really important question, because for a lot of the work on plant invaders, so there's been a lot of interest in this idea that plant invaders appear to be phenologically early, and they appear to be tracking climate change. And I've done a lot of that work. There's strong evidence that invaders appear to phenologically track climate change better. But there's lots of other things that might co-vary with phenology, so it might not be the key trait. So Deirdre's work is going to be critical to trying to unpack the mechanisms behind some of these correlations that we're seeing.
Manning Park had a lot of interesting benefits. As Lizzie mentioned earlier, we were interested in looking at geographic scale. So we have transects all the way up to Smithers down to Manning Park. And we were really interested in finding sites that had similar communities and had similar species assemblages. So we had a long list of species that we essentially went on a scavenger hunt and tried to find which species were present in multiple areas, which ones matched other data sets that we had. And Manning Park was just such a beautiful place to work. It was accessible, and it offered everything we needed in terms of the ecosystems we were looking for.
Yeah, Manning Park has been great to work in. Because phenology, one of the cues is day length, we want to be able to address it across a geographical gradient. So we can look at whether there's differences in how a very northern population from Smithers might respond differently to day length than a population 5 degrees south of there, which is about Manning Park. And so we were hoping Manning Park would work. The first time we ever visited was the summer of 2018 when I was on like a 2-week field trip across potential field sites for BC. It was one of the staff for the summer came out and sort of showed us around and helped us look for some of the species that we wanted to find. And that was a really great trip. I think I looked at like 6 or 7 different, maybe 8, many sites. And so Manning and Smithers were the 2 we actually did experiments at. So they're 2 of our linchpin sites.
So I think for me, the most interesting experience was just how intimately you get to know the ecosystem and how that influences how you interpret your data and how you think about the species. Even within a few weeks, I could look off into the distance and know the vague shape was the species I was looking for. And so it was really cool to get an understanding of where would we find these species, who would they be with, and what kind of key habitat traits might we hone into if we're looking for them. And you just don't get that kind of experience when you're sitting in your lab or reading a paper. So for me, it just kind of highlights how unique and special these systems are and how much we don't understand. How is it going to change? What is this forest going to look like in 30 years, 50 years, 100 years?
And for me, I guess I have 2 things. One is I've always loved larch. I figured out what larch was very late in life, I think. I found it when I was like in my 30s in Europe and was like, oh my gosh, that's such a crazy species. It's like a conifer, but it looks like it's gonna drop its leaves. And for someone who studies phenology, larch is super cool because we actually mainly work on angiosperms, partly because they're deciduous, which means we can put them in chambers and get them to leaf out in a predictable way that we understand. And there's a lot less research on the phenology of evergreen or coniferous species because they keep their leaves all winter. And so larch is this amazing thing that I would just love to study, and I just find it like a very cute Muppet-like plant that I'm just like fascinated by, and it's obviously so beautiful in the fall. And Manning Park has this random population of subalpine larch, and it is one of the more eastern populations that you can find. And so I would love someday to study those. As a person, I'm not a botanist, but I study plants. So as someone who loves plants, they're so exciting because you don't see larch very often on this side of BC, and they're this magical little population hanging out on Mount Frosty. They're super cool. The other experience I had was when we first started working here, we agreed to work at Manning Park in 2018. And having not ever tried to work in like the mountainous areas of BC, I wasn't sure when we should start collecting, but Deirdre and I were troubleshooting. We're like, May seems like when you would go out and start cutting up plants and measuring leaves and tagging things. And I can't remember why, if I was scouting it, but we were thinking early May is when we would go, and I ended up in Manning Park in early May. It's definitely not spring-like in early May. It's like covered in snow and ice. Everything is a twig. Nothing is leafing out.
Even when we went in late May, I soon realized I had to learn the winter ID, which is a whole other set of traits, and you don't have the pretty flowers and the leaves. But that was a great experience, and it just shows that you have to sometimes adjust and when you're doing field work and roll with things.
Yes, I think I've definitely learned that ideally at Manning Park you would sample in late June or later. That would be a good time to go and work on plants. And really the earliest probably is sometime in June. You can make do, but if you want to go see the plants in full flush, you should wait till sometime in later June.